Hi student, today we are going to see about differential staining method. There are two types of differential staining method. One of gram staining and another one is acid form staining. First we will see gram staining. The gram stain developed by Christian Gram in the 1884 was the first differential staining technique in use, and it is still an important tool for distinguish between two main types of bacteria for gram positive and gram negative. Principles are gram staining. Gram positive bacteria retain crystal violet and appear deep violet in color, and gram negative bacteria lose the crystal violet on decolorizing agent, that is acetone or alcohol, and are counter stained by diluted carboxylation and appear pink in color. Iodine is used as a modern stain. It increases the interaction between the cell and the stain so that the cell is stained more strongly. Mechanism of gram staining. The cell wall of gram negative bacteria contains a higher percentage of lipid content than gram positive bacteria. The alcohol treatment extracts the lipid. This result increase in porosity of the cell wall. This crystal violet iodine complex can be extracted and the gram negative organisms is decolorized. The cells take up the color of the counter stain, diluted carboxylation. The cell wall of gram positive bacteria have lower lipid content and hence the pore size is small. Crystal violet iodine complex cannot be extracted. Therefore, gram positive cells remain violet in color. Procedure of gram staining. Place the slide with heat fixed smear on staining tray. Gentle fluid smear with the crystal violet. It is a primary staining and let stand for 1 minute or 60 seconds. Tilt the slide slightly and gently rinse with the tap water or distilled water. Gently fluid the smear with grams iodine. It is a modern stain to attraction between the cell and dyes. And let stand for 1 minute. Till the slide slightly and gently rinse with tap water or distilled water. The smear will appear as a purple circle on the slide. Next we will using decolorizing agent acetone or alcohol. The alcohol to remove the crystal violet iodine complex from gram negative cells. Since crystal violet did not adhere strongly to the thin peptidoglycan layer and the cells become transparent. Till the slide slightly and apply the alcohol drops by drops for 2 to 3 seconds until the alcohol runs almost clear. Be careful not to over decolorize. Immediately rinse with water and gentle fluid with saffron or carboxylation. It is a counter stain. It is cationic in nature and let stand for 30 seconds. Tilt the slide slightly and gentle rinse with tap water or distilled water using wash bottle, and the slides are air dried, and view the smear using a light microscope under 10x, 40x and 100x oil immersion. The cells will appear, gram positive organisms will appear purple in color and gram negative organisms will pink in color. This is the microscopic view of organisms to differentiate the gram positive and gram negative organisms. Next differential staining method is a acid form staining. Acid form staining is another important differential staining procedures. Acid form staining is also known as a Sjctnc-Mrs staining. Used to identify specialized bacteria that have waxy metabolic acid in their cell wall. Principle of acid form staining. A few species, particularly those in the genus Microbiology-Unit, do not bind simple stain and must be stained by heating with mixture of basic functions and phenols. Once basic function has penetrated with the aid of heat or phenol. Acid phos cells are not easily decolorized by decolorizing agent for sulfuric acid and hence remain red or pink in color. This is due to the high lipid content of acid phos cells. Particularly, mycolic acid is the responsible for acid phosness. Non-acid phosed bacteria decolorized by decolorizing agent, that is decolorizing agent we are using sulfuric acid, and are stained by counter stain methylene blue. This method is used to identifying Mycobacterium tuberculosis and Mycobacterium leprae, the pathogens responsible for tuberculosis and leprosy respectively. Acid phos bacteria appear pink in color, whereas non-acid phos bacteria appear blue in color. Procedure for acid phos staining: suspension of tubercle bacilli smeared on a clear glass slide and heat fixed. Apply bright pink dye for using xyl corbel function on smeared slide and placed over a water bath for 3 to 5 minutes, and next rinse the slide for running tap water, and we are using decolorized with 20% diluted sulfuric acid for 1 to 2 minutes. Lastly, counter stain. It also cationic in nature. Using methylene blue is applied for 1 to 2 minutes and slides washed with water and air dried. After dried the slides observed under microscope. Acid phos waxy bacterial cells are bright pink and non-acid phos bacterial cells are blue in color. These are showing microscopic view of non-acid phos bacteria will appear blue in color and acid phos bacteria will appear pink in color. First we will adding primary stain, corbel function, and application. We are using the mordant in acid phos staining. For using heat is the mordant. To fix the stain, interaction between the cells and dye, we are using heat is the mordant, and decolorizing agent for sulfuric acid 20% and counter stain for methylene blue. The cells will appear. Acid phos staining is a pink in color and non-acid phos staining is a blue in color. Thank you, children.